purpose of this video is to give you some clues about how to handle the acids, bases and pH buffers lab. The first thing you should do is take a look at the background. It gives some interesting information about buffers, acids and bases, and also about buffer capacity as well. And all of this is stuff that you'll be investigating while you're doing the procedure here in the lab. So the first thing you'll have to do is an introduction to pH indicators. Specifically bromphymol blue, I think, is what you'll be using. And what you'll do is you'll get 6 test tubes, and you can just lay them out here, and you'll notice that they get numbered as you put them down. So you can mouse over and you'll see it has a separate number. Just lay them out in order. In the first one you're going to put water. I'm only going to show you the first one here, but this is pretty much how you do it. You get the water, put it over the test tube. It tells you exactly what you need to do, and you'll add here 10 mils, so go ahead and do that. And you'll see it fills it up. And then the idea is to get a pH meter from the instruments area, and you stick it over the test tube, and it will come up with a reading. And there's the reading on your test tube, and you can always go closer to get a better idea of the reading. And you'll do that and repeat it for the rest of those. At some other point, just a little bit later on, you'll be doing this bromphymol blue. So you get the bromphymol blue, you stick it over the test tube. It says here to add 2 drops, so we'll add 2 drops and we'll hit OK. And you can get an idea of the colour. Remember, zoom in if you want to see the colour a little bit better. Alright, so you'll do that for all 6 of them. We'll clear the bench, and we will continue on with the rest of it. Next, we're looking at the phosphate buffer system. In this case, we're going to need 4 solutions. One's going to contain water, one has sodium monobasic phosphate, and the other one has sodium dibasic phosphate. Now these two are completely different. When you combine the two, that's where we get a buffer, and that's a weak acid, that's a weak base. So when we combine the two, that's where we get a phosphate buffer. So we'll go ahead and follow the instructions here and put 5 mils of each of these solutions in the first 3 and 2.5 mils of each of the monobasic and dibasic phosphates in test tube 4. So we use the containers, of course, and we'll have 4 of those. And we'll go get the materials. Do the water first, and we need 5 mils of that, and then we need 5 mils of the monobasic phosphate. So just sort of mouse over here, and you see that's the dibasic, here's the monobasic over here. They put it far away from the other one so you don't get them mixed up. Just make sure you go over the test tube you want, and again add 5 mils. The next one is the dibasic, put that in here. And the last one is the combination of the two. So we'll get the dibasic here, add 2.5 mils. And the monobasic over here, and I'll add 2.5 mils of that as well. Alright, so we've got 5 mils of solutions in each of these, and we're good to go. So we need a pH meter in each of them. And you can see all the pH readings for each of them there. Now I'm just going to show you the response to the hydrochloric acid for one of these, and you'll have to do it for all the others so you can see the results here. But what you're doing is you're measuring the pH after you add the HCl, and you're going to see which one of these is most affected by the HCl. Some of them may not be as affected by the HCl as some of the other ones will be. Alright, so we've placed one pH meter onto each of the test tubes. We've done that. Record the contents of each solution's pH. We've done that. So you need to put that into your lab notes. All of that will go in there. Then what you'll do is get a clean 50 ml beaker from the container shelf. And then you'll add 10 ml of 0.5 molar hydrochloric acid. There we go, that's 0.5 molar hydrochloric acid. So I'll put that into the beaker. Remember, you've always got to put it over the beaker if you want to add, right over the beaker if you want to add something to it. There you go. Take a dropper from the container shelf. Here's a dropper. And we'll stick that into the container. And you'll notice it fills up. So now what we can do is we can take it over and put 2 drops in the first test tube. So put it over the test tube and then add 2 drops, hit OK, and you can see what the pH does. Now you'll repeat that for each of these and see what the pH does in each of those instances. After you've done that, you will repeat this whole setup for the addition of sodium hydroxide. So you'll have to clear off the bench and reset these up. You can't just go ahead and add sodium hydroxide to each of these as it stands because they've already had hydrochloric acid added to them. So clear off the bench, set it up again, and then do the sodium hydroxide. It's pretty much the same situation as we did for the acid. So I'm going to clear this off now and we'll move on to the next part. We're now going to look at the buffering capacity of a pH buffer. So what we'll do is we'll just follow the instructions. Place the test tube, add 2.5 ml of dibasic phosphate and 2.5 ml of monobasic phosphate. So we're actually making a buffer here because we're mixing those 2 phosphates together. There's our dibasic. So we need 2.5 ml of that one and 2.5 ml of the other one. Hit OK. Place the pH meter. It'll tell us what the pH is of the buffer solution. Make sure you record that. And then place a 50 milliliter beaker on the bench. And then you will add 20 ml of the 0.5 molar hydrochloric acid. Again, we get a dropper, and we stick it into the hydrochloric acid. Alright, so it fills up. Now what we're going to do is add this to the test tube, but we're only adding 1 drop. And then you'll see that the pH will change. So in our lab notes, here's what we'll put down. We'll say 0 drops. That's initial. The pH was 7.21. And then after 1 drop, and this is for HCl, and I should probably do that up here. We'll say HCl. The 1 drop, it was now 7.12. So I'll go ahead and add another drop and we'll get the pH again. So now it's 2 drops, that's going to be 7.03. And I'll do again for 3 drops. And of course you'll see that the pH is dropping here. Alright, so that's all I wanted to do for the acid. You can do the rest for the base. I'll save the notes here, and we'll go back to the procedure. And you will you'll need to repeat this procedure for the sodium hydroxide as well. And I'm thinking that you will need to repeat the whole thing for the addition of base, not just from here. You'll have to clear off the bench and start this process again for the addition of base. And what I'm going to do is I'm going to do the addition of 3 drops of base as well, so that I can in the next video explain the math involved for doing the graph. Alright, so we'll do this again here, and we'll get the materials. We'll do 2.5 mils of this. Now of course you'll be doing a lot more drops than I'm doing. I'm just doing this so that you can get an idea of what you need to be doing. Now we're just repeating the process, except now we're going to use sodium hydroxide instead. There it is. We'll put 20 mils in there. We'll get dropper. Don't forget the pH meter, we stick that in there. And again we'll start out at 7.21. And we will now add the, make sure I fill that up. Yeah, it looks like it's full already. Let's take it over here and we'll add a drop of base. And you'll see it goes up to 7.29. Now remember, you will be adding a lot more than I'm going to be adding here. So 1 drop, 7.29. Of course it's going up because we're adding base. I go take this over here and I add another drop, 7.38. And I'll add one more. And 7.47. That's for 3 drops. Alright, so this will help us with the graph when we get to that part of it. Now save the notes, move back to the procedure. So that's pretty much it for that, and I will do another video that shows what you need to do for the actual lab report.